Hey guys, welcome back. In this set of notes, I am hoping to spark your imagination and teach you a little bit more about protein synthesis. Now, protein synthesis is one of the big processes that we have to know in biology. In fact, protein synthesis is part of that central dogma of biology that we talked about in class. So, protein synthesis simply means the process by which a protein is made. The word synthesis means to make. So protein synthesis means to make a protein. Now, in protein synthesis, there are two steps. Step number one is called transcription. Step number two is called translation. So let's write down some of the key things that we need to know about both of these processes. So first up, let's write down where these processes happen. Transcription happens in the nucleus of a cell, while translation happens at a ribosome in the cytoplasm of a cell. Next, let's talk about who the molecule is that does this. So in transcription, this uses a molecule called RNA polymerase. RNA polymerase puts the new strand of RNA together. In translation, that mRNA strand is going to be translated by the ribosome, and it's going to use ribosomal rRNA to help. So ribosomal RNA, remember, is abbreviated little r and then big RNA. So rRNA or rRNA. Just 2 R's, not 3. So what's happening in these processes? Well, in transcription, we are taking DNA and we're going to use DNA to make mRNA. Remember, mRNA stands for messenger RNA. In translation, we're going to use that mRNA, and our goal is to make a protein. All right, so when does this process happen? Well, these processes happen whenever the cell needs a protein. So both processes have to happen with each other. And so transcription is going to happen first, and translation is going to happen second. They can never go in the opposite order. Okay, so a couple other things we need to know here. First up, with transcription, we need to make sure that we understand that DNA cannot leave the nucleus. So we're going to put that in a Y section. So DNA cannot leave the nucleus. So we make a copy of the DNA called mRNA that can leave the nucleus. Alrighty. For translation, it's a little bit different. For translation, we need proteins. So mRNA is matched with tRNA, and tRNA stands for transfer RNA. tRNA carries amino acids, and those amino acids are going to connect together or bond, which means to stick together, and remember, amino acids are the building blocks of a protein. So it carries amino acids which bond together to make a protein. In class, we're going to model how that process happens in a cell. But for today, I want to focus on you being able to use DNA to make an mRNA copy, and then I want you to be able to use the mRNA copy to make a protein chain. And I want you to be able to do this on paper. Okay, so a couple things you have to know in addition to this. Remember that you need to know mRNA, you need to know tRNA, you need to know rRNA, you need to make sure that you know the word transcription, translation, and then a couple words that we haven't talked about yet that you need to know are codon. We're going to talk about this one in just a minute. And anticodon, which we will talk about a little bit later next week. I'm going to put mRNA next to codon because you'll find codons on mRNA, and anticodon is going to have something to do with tRNA. Then, of course, you need to know amino acid. You need to know protein. And you need to know the word gene. Alrighty, so let's talk about gene in a little more detail. We talked last semester about chromosomes. And we drew chromosomes something like this. And remember, a chromosome is a strand of DNA. Now, if this chromosome were doubled, that would be the X shape, but we're just going to deal with one. So if I were to zoom in on just this section of my chromosome, I would see something that looks like this. This is that DNA double helix. Then if I were to look at just this section of the DNA, that is a gene. Here's another section of DNA. That is a gene. So genes are segments of DNA that code for a protein. Different proteins have different genes. Okay, so let's talk about how we get from a gene to a protein. Now remember, when we're doing protein synthesis, we need to start with transcription. and transcription happens in the nucleus. So I have a gene written here on my paper in DNA, and that's in the nucleus. The first thing that I need to do in transcription is I need to convert that DNA to make mRNA. When you learned about mRNA, remember that mRNA uses U's instead of T's. All right, so we're going to use this strand of DNA to make a strand of mRNA. Now, if we're pairing up DNA, remember apples are in the tree and cars are in the garage. But that's a little bit different with mRNA. With mRNA, because T is used in, or because U is used instead of T, the A will pair with U. T in DNA will still pair with A. And then if there's a C in DNA, it will pair with G in mRNA. And if there's a G in mRNA, or I'm sorry, a G in DNA, it will pair with C in mRNA. Okay, so here's what we do. Here's a T in DNA. We know that T pairs with A. So we'll put an A for mRNA. We're going to complementary base pair. We put the opposite base. We have an A in DNA. Now remember, we learned before in DNA that A pairs with T, but RNA can't have T's. RNA has U's, uracil. So this A pairs with U. C pairs with G, just like always. T still pairs with A. T with A. T with A. All right, pause the video and see if you can finish base pairing the rest of this strand of mRNA. When you finish, hit play on the video to check your answer. Welcome back. You should have finished pairing your mRNA, and you should have written G pairs with C, A pairs with U, G with C, C with G, G with C, A with U, A with U, T with A, and C with G. Now we have a single strand of mRNA. This mRNA is now going to leave the nucleus and it's going to go to a ribosome. Remember, this is in the cytoplasm. Now, a ribosome looks kind of like a hamburger bun. It's got a big top and a little bottom half. And this little strand of mRNA is going to go into the ribosome just like this. And this is where tRNAs are going to pair up to make a protein. But on paper, we do the process a little bit different. All right, so I'm going to rewrite our mRNA strand real quickly. Okay, now mRNA is divided into words. In the English language, we can have words that are anywhere from 1 letter long to 10, 12 letters long or more. But mRNA is great because mRNA is always divided into 3-letter words. And those 3-letter words are called codons. So what I'm going to do is I'm going to draw a vertical line in front of the first letter. Then I'm going to count 3 letters over, 1, 2, 3, and draw a line. This AUG is a codon. So a codon is 3 bases that match 1 amino acid. So these three bases matches one building block for a protein. All right, so I'm going to keep counting. 1, 2, 3. AAA is another codon. So here's a second building block, a second amino acid. 1, 2, 3. CUC is another codon. GCU is the fourth, and UAG is a fifth codon. So this strand of mRNA has 1, 2, 3, 4, 5 amino acids because it has 5 codons. Alrighty. So how do we know which amino acids go with what? Well, in a cell, tRNA would be used, but we don't just have tRNAs floating around our paper. So instead, we have to use a codon chart. Now, there are two types of codon charts that we can use. The first codon chart that we can use is the grid style chart, and it looks like this. You have a copy of this that's yours to keep, so make sure you put that chart in your notebook so that you have it for reference. This particular chart in the middle here is where we are going to find the amino acids, and this chart has abbreviations. Some will have the entire word written out. But all charts are set up the same way. They have first base on the left, second base on the top, and third base down the right side. All right, so here's how we read this codon. So we start with the first base. The first base in our first codon is A. So I'm going to look at the 4 letters on the left side. Since my first letter is A, I'm going to look at this row that has A on it. So my amino acid is somewhere on this row. All right, my next letter, whoops, marker doesn't want to go away. Next, excuse me, next letter is U. Since that's the second base, I go up to the top where it says second base, and I find the U across the top. So that's right here. Excuse me, sorry about that. All right, so I'm going to look just at the U column. Now I want you to notice that there is a box where the A and the U cross, and that is right here. This box is where your amino acid is going to be, wherever your first and your second letter cross. Alrighty, our third letter is G. So to identify which of these amino acids, I go to the right side where it says third base, and I find the G. Now notice that G is printed 4 times. The G that you use, or the letter that you use, will be on the same row of your first letter. So we highlighted A here, so we're going to use the G that is within the A box. We're going to go across, and that means that this is our first amino acid. So underneath our mRNA, we're going to write this abbreviation MET. In a cell, MET will always be the first amino acid in a protein. So just know that in real life, MET is always first. However, for practice problems we give you, it may not be. So you always want to double check it. All right, so let's do the next base together. Let me grab a fresh codon chart. I would suggest yours that you don't draw on it. I'm just drawing to show you how this works. Alrighty, so our first base is A. So again, I'm going to go to the A row on the first base side. My amino acid is going to be somewhere in here. All right, second base is A. So I go to second base on the top, and I choose the A across the top. So since this is the A column, this is where A and A intersect. So it's either going to be ASN or LYS. How do I know which? Well, I look at the third base. Third base is A. Remember, the third base is on the right side, but I pick the one that's in the row of the first base. So here's that A. So I come across, and my second base is going to be LYS. So I draw a line that represents a bond, and I write LYS. All right. So I want you to try CUC on your own using your grid codon chart. Pause the video. When you think you've got it, come back and check your answer. All right, welcome back. So you should have found C, U, C. So first letter C, second letter U. So it's somewhere in this box here, which really only gives you the same choice. But just to double check, look for that C here. And our next base has to be LEU, which stands for leucine. Okay. You do not need to memorize any of these abbreviations. You do not need to memorize what any of these things stand for. You just need to be able to use this chart. Okay, so that's the grid chart. But remember I said there are two different charts you can use. So the other chart that you have the option of using is the Bullseye chart, and it looks like this. Now I think this one is a little bit easier to use, and it's pretty simple. You start in the center, and then you work your way out. So the center ring is the first base. The next ring out is the second base. And the last ring all around here is the third base. And this chart is divided into quadrants. So you have a vertical line and a horizontal line that divides it into a G quadrant, a U quadrant, an A quadrant, and a C quadrant. Okay, so what does this look like? Well, let me show you. Our next codon is GCU. So we're going to start with our first base, which is G. So I'm going to go to the center, and I'm going to find G. Now I'm only going to work in the G quadrant. I can ignore the rest of this chart. So now I go to the second base. The second base on here is C. So I'm going to find the C that's next to the G. So that's right here. So I come to this level. Then I look at my third base, which is U. So from the C row, I look for the U, which is right here. So that means that my amino acid is whatever's on the very outside. And that happens to be ALA. So I write that in. All right, so pause the video and see if you can use your circle chart to figure out UAG. Welcome back. All right, so let's look and see if we can figure out what UAG is together, or you can check your answer. So for UAG, I start in the center with U. I then find the A that's connected to the U, which is right here. Then I find the G that is connected to that A, so that's right here. So I have a stop. Now, stop is not an amino acid. It literally is like a stop sign. It just tells the ribosome to stop making the protein here. So the protein is MET, LYS, LEU, and ALA. A lot of times you'll see this drawn with little circles. So MET, LYS, LEU, and ALA. So this whole entire thing is what we call a polypeptide chain. Also known as an unfolded protein. So this is a protein, it just doesn't work yet. It'll have to be folded up later in a process that you'll learn in AP Bio or in college that makes this protein work. Now you'll notice that in between the amino acids, there are, let me switch colors real quick to something that's a little brighter. How about purple? There are these little bonds. These bonds that hold the amino acids together are called peptide bonds. So this is what connects the amino acids. All right, so thank you for sticking with me through this part of the lesson. Couple of reminders for you as you're doing these. If you see a strand of nitrogen bases that has a T in it, that's going to be DNA. If you see a strand that has U's in it, that's mRNA. So remember, when you have a strand with T's or you're writing a strand of DNA, you have to use T. When you have a strand with U or you're writing mRNA, you've got to use U. Remember that transcription takes DNA and makes mRNA in the nucleus. Then that mRNA leaves the nucleus, goes to a ribosome, where it is then used to make a protein. So the protein that is made is called a polypeptide, and it's connected with peptide bonds. All right, if you have any questions, let me know. Otherwise, you can now start your practice activity that you were assigned in class. Again, thanks for sticking with me and have a fantastic day.